Hi, Michelle Glass here. We are ready to continue our Chapter 6, Lecture 6 with Episode 5. And our topic here is that final step in the process of a cell making ATP from glucose. So we started with the step of glycolysis, which is occurring in the cytosol of the cell. That's the breakdown of glucose into pyruvic acid molecules, ATP, and also NADH. That pyruvic acid moves into the mitochondria where it's oxidized. So we looked at pyruvate oxidation. That gives us the 2-carbon acetate, which is paired with the coenzyme A to make acetyl-CoA. That is happening in the matrix of the mitochondria. And then from there, we saw those acetate molecules moving to the citric acid cycle, where we are still inside the matrix of the mitochondria, and we see here that the oxidation of the acetyl-CoA is going to give us additional NADH molecules, FADH2 molecules, and also some ATP. And now we are ready for our final step, which is the electron transport chain. You can also call it electron transport system. And then finally, the actual production or phosphorylation of our ADP molecule into our ATP molecule. So we will, at the end of all this step, finally get our output of ATP. Okay, so here we are still in our mitochondrial matrix looking at, again, that process of citric acid cycle. So we have produced here some NADH, FADH2, and also some ATP. We are ready to look at our electron transport chain, which is going to be in that mitochondrial inner membrane. Remember, our NADH molecules and that FADH2 molecule is acting like a taxi cab or an Uber. It's bringing those electrons and, really electrons. It's bringing those electrons to the electron transport chain. So total from all of the reactions that we've looked at so far, we've produced a total of 10 NADH molecules and 2 FADH2 molecules. We are going to see that our final steps are going to be taking place in the inner membrane, and also we're going to be utilizing that intermembrane space. So that's what we're going to be, that's where we're going to be in this process. Okay, so when our NADH and our FADH2 are bringing our electrons to the inner membrane space, really we have hydrogen atoms coming here, and the hydrogen atoms are being split into those electrons and those proton components. So notice that we don't just have electrons being brought here, but we also have hydrogen ions, which are protons essentially. What we're going to see happening is the electron is going to be passed from our taxi, from our Uber, to our first electron acceptor. So remember, as we're passing our electrons down the chain, little bits of energy are going to be released. The energy that is released from this movement of an electron will fuel a hydrogen pump. And so the hydrogen is going to be pumped into that intermembrane space. And then those electrons are continuing through the chain. So the electrons pass to the next the next electron acceptor. And as that electron moves, there's energy released, and there's hydrogen ion being pumped into that intermembrane space. And that's going to continue. So what we see happening is we're setting up a really high concentration of hydrogen ion in the intermembrane space. Very little, if any, is there in the matrix. Those electrons have been passed from one electron acceptor to another until we get to our final electron acceptor, which is, of course, our oxygen molecule. So this whole process we have described as aerobic respiration, meaning oxygen is required. We have seen respiration is our term, which means gas exchange. So we've talked about where we produce the CO2 molecules and how that's the reason that we are exhaling CO2 gas. This is the reason that we are inhaling the oxygen, right? Because we need it here to grab those electrons at the end. that electron is super attracted to those oxygen molecules. And so that's really the thing that's pulling those electrons down step by step by step. So remember, our oxygen is our final electron acceptor. Now, again, we've seen that those electrons moving down the electron transport chain gave us the energy we needed to pump hydrogen into that intermembrane space. Once we have created that super high concentration of hydrogen, we can then send it through this really special enzyme called the ATP synthase. So this is an example of chemiosmosis because we are moving our hydrogen from a high concentration to a low concentration. It's moving specifically through this ATP synthase enzyme, which is a really cool enzyme because when the hydrogen comes in, it actually is causing it to spin around. And this spin that we're achieving in the ATP synthase molecule gives us the energy we need to make our ATP. When you add a phosphate to an ADP, we can say that we're phosphorylating adenosine diphosphate, creating our ATP, our adenosine triphosphate. So because you have used energy pulled off by oxidizing molecules in each of our procedures, starting with glycolysis, pyruvic acid, oxidation, citric acid cycle, we've basically pulled off those electrons through those processes of oxidation, and those electrons are used here at the electron transport chain to phosphorylate our ATP. We're going to call this whole process of creating the ATP here oxidation phosphorylation. Notice too, as that hydrogen is coming through the ATP synthase, it's pairing with that oxygen so that we don't have just electrons and protons separate in the cell, but we have a stable water molecule produced at the end. Again, oxidation phosphorylation is the description of what's happening here at the electron transport system, and including that chemiosmosis, that movement of the hydrogen ion through that ATP synthase. Because again, the energy that we have fueling this whole process, it was coming from oxidation of glucose and then pyruvate and then acetate. We can describe this process as oxidation phosphorylation. At the end of this process, this is where we're getting the bulk of our ATP production. Right here, we usually get maybe 32 to 34 ATP molecules being produced. If we look at what we've already made in our ATP molecules, notice that we've already made 4. So if we make 32 ATP at the end of the electron transport chain, 32 plus 4 is 36. If we are making 34 ATP at the end of our electron transport chain, then we would have a total of 38 ATPs being produced. So there's a variation depending on, lots of different factors in the cell, but typically we talk about in total glycolysis. We don't make any ATP at pyruvate oxidation, so glycolysis and citric acid cycle, and then what's happening here in oxidation phosphorylation, we typically talk about collectively making 36 to 38 ATPs from a single glucose molecule. So that is a number you should know, 36 to 38 total ATPs made at the end of all of this process. Okay, and that is it for now.